Thank you for the great presentation there before. I would like to take a few minutes before we get started. We're going to discuss a few things on contemporary energy research. And rather than having a panel discussion, I thought it would be good if we had just had a few of our colleagues present some of their work, just to give you an idea of the breadth that is going on here at the Energy Initiative. But before starting that, I'd like to thank Bob and wish him all the best for his supposed retirement. All the evidence suggests that he is working just as hard as ever, but for a fraction of his previous salary. But he will now have ample time and opportunity to take time off to spend with his lovely granddaughter on the West Coast. So we wish you all the best, Bob, on your retirement, and look forward to seeing you on campus a lot, even though you are retired. On a personal note, it has been an honor and a privilege to have you as a fellow Wisconsin alumnus, a department head, the Energy Initiative director, and friend. Thanks for everything you've done for MIT over the past 50 years, and we really look forward to your continuing engagement with us. Thank you. As I said, today's session will discuss just 5 of the many different areas in contemporary energy research facilitated by MIT. It's again to emphasize the breadth and the depth of contributions from our colleagues and just some of the contributions that we can anticipate. So I was going to talk to you a little bit about some of the work that I've been doing here on selective CO2 removal through electrochemistry. And the reason being, of course, is that global warming and the global warming is a major issue and it's become all the more evident over the last year or so when we've had significant increases in flooding, fires, storms, et cetera, et cetera. And if we look here, what we see is that there is a, should have been, does anybody have the clicker? Anyways. If not, we recognize that there has been a major increase in temperature over the last 40 years or so in the environment. And this is increasing at a rapidly growing rate. At the moment, it's well over 1 degree Celsius rise. And if you look at what it is, it's basically been directly related to the actual amount of CO2 that's been emitted. If you look at the temperature rise as a function of the cumulative CO2 emissions over the decades since the dawn of the industrial age, of course, we recognize that, thank you, that we had about 288 ppm. We are now at 420 ppm. If we continue going at this rate, I have no idea what it would be in 2050, but pretty significant. And the temperatures there would be on the order of 2.5, 3 degrees, about 2.5 degrees above the pre-industrial level. These are major issues and they need to be addressed. So what we want to do is think about where did the CO2 come from, what is happening to the CO2 in this region. So basically, if we look at the total emissions currently on the order of about 35 gigatons per year of CO2 coming from fossil fuels and industry and from land use change. So we get about close to 40 gigatons per year of the CO2 emissions. On the other hand, these are also somewhat mediated because we have a reabsorption by the terrestrial activity here, but we also have the ocean as a major sink for the CO2, which sounds like it might be a good opportunity to remove some of the CO2 from the environment. But of course, what this also means is that we lead to significant acidification of the oceans that needs to be ameliorated as well. And this means that we get an atmospheric CO2 growth rate of around about 20 gigatons per year. And this is something that we cannot sustain, and we need to find ways of addressing this. Of course, what we've got to do is intercept the emissions and also see if we can then remove it from the atmosphere as well in direct air capture so we can minimize the accumulation and therefore the global warming effect. So the way in which we go about capturing the CO2, I think it's well understood, is that what we have some kind of a base sorbent, and we react it with CO2 to form some complex as shown here. What we want to do, once we've loaded this up, we need to sort of break this apart, break this bond, capture the CO2 as a pure stream, and regenerate the base so we can reuse it. And of course, this is normally done through thermal means. The typical breaking of this bond is done in a desorber in the bed here like this by thermal energy means. So we're really heating things up quite dramatically up to about 120 degrees, maybe 100 degrees, 120 degrees, which means we need some significant heat integration. We need thermal and regeneration sources and things like that. So our idea is, can we actually work on using electrochemistry so we can use more renewable resources, and also can we keep it at isothermal type operations. So this is basically the concept here. We want to capture and release CO2 by regulating the activation and deactivation of certain species and applied potentials governed by a Nernst equation. This is a Nernst equation. But what it really says is the amount, the fraction of the species that is now activated or reduced or activated is a function of the applied voltage of applied potential in this particular case. And indeed, if we get sufficiently negative potentials, what we can get absorption of the CO2 and form the complex there. But if we want to desorb it now, we've got to break that complex. We need to go to a much more positive potentials in this area and get the desorption. And the reason this is really good is because the energy is targeted to the sorbent molecules only. We're not heating and cooling large quantities of sorbent material. We're only directing at the actual complexing agents themselves, which has some advantages. The processes can be modular, compact, isothermal, plug and play, and driven by renewable energy. So there are different ways in which we can do this that we've been investigating. The first is a direct complexation process. And what we have in this particular case, some molecule, in this case it's naphthoquinone, but there are many others we could use. We can bring that to a cathode where we actually do the reduction at this particular point. We reduce this and activate it, so it becomes a very strong a nucleophile for the CO2. The CO2 will then react with it to form a complex, as shown here. This carbonate complex is shown in this particular case and be trapped in that regard. When you want to release the CO2 and regenerate the sorbent, we take it to an anode where we do the oxidation and it comes back to the neutral quinone, which has no affinity for CO2, and CO2 can be pulled off. And the next stage would be an indirect approach in a protic-type environment in which you've got water or whatever. And here what we have is a quinone that when it is reduced, it basically absorbs the protons from the aqueous solution and not the CO2. But what that does is raises the pH, which allows the CO2 to be absorbed here as a bicarbonate. And when you want to go through the regeneration and recover the CO2, we do an oxidation here. When you oxidize it, it's releasing the protons, which reduces the pH, which means we change the speciation of the bicarbonate to pure CO2, to CO2 molecules, and that can then be drawn off through the gas diffusion electrodes in this particular case. And then there's a final case which we have is an indirect but displacement. In this particular case, what we do is actually take an amine, say an amine and CO2, and complex that. And what we do when we go to the anode chamber, what happens is we release copper ions from the electrodes. These copper ions complex preferentially with the amines and displace the CO2 is now reduced, is now kicked out, and it comes off as a pure CO2 stream. I'll show you another diagram of that in a moment. So the implementation of these processes requires looking at identifying and optimizing the redox agents we need in these systems. We want to be able to optimize the system so we have low voltage swings and therefore minimize the energy requirements in the system. We want efficient contact and cell designs, and then coupling with non-carbon energy sources such as solar, wind, and nuclear type resources. So the first case is shown here. I don't have much time here for this. The first case is taking I want to just discuss this one here, in which we take the amine, the CO2, it complexes CO2 to form the carbamate in the absorption column. Once we take that carbamate solution to the anode chamber of the electrochemical cell, we release cupric ions that come in there and complex with the amine and kick off the CO2 that can then be taken off in a flash tank. We then take this complex to the cathode side and be able to plate out the CO2 and regenerate the amines and send them back again. It turns out that this looks like a very energetically favorable process, and it seems to have some very significant opportunities relative to traditional thermal type processing. So there are others we've looked at using quinone type systems that we've shown that we can do in a sequence way. And we've actually taken this result and formed a company, Verdox, that has been recognized with the $1 million X Prize Carbon Removal Award last year. There are other ways we can do this using indirect pH swing that can take a continuous stream of air, rich air, CO2 rich air that would be from the ambient environment, and then resulting in a pure CO2 stream. And we get a cumulative CO2 removal from ambient air, which is really a successful application of these kinds of approaches. And that'd be good for direct air capture type applications, obviously. And direct air type applications, the nice part about these kind of electrochemical processes is that while the actual capture units can be very, very large and occupy large areas, large swaths of land, the actual units themselves, because you're using very concentrated redox agents here, they can actually be used to capture the CO2, concentrate it quite dramatically, and then just be able to do regeneration in a much smaller unit over here. So that provides a lot greater incentive for doing this. I won't go through this right now, but I just want to say that basically these electrochemically modulated CO2 capture operations have significant advantages. They're isothermal. There's no external heat sources are needed for these systems. They can be plug and play. They can be put in anywhere. You can get a good electrical source. They're modular. They can be easily scaled out. They're favorable energetics, 1 to 2, maybe 3 gigajoules per ton of CO2, depending on the conditions, and they can use non-carbon energy sources. They're applicable across multiple scales and concentrations. We can look at power generation, industry, mobile sources, indoor environments, direct air capture, direct ocean removal, etc. But we do, of course, need to implement them in some ways and optimize these systems, which provides a very rich research area target. So with that, I'd like to thank Bob and Mighty who have all played a major role in enabling development of these technologies, and thank you. But in the meantime, what do we do with the CO2 we captured? You know, that is a big issue. And this is where Yogi is going to tell us what he's done, because we've got direct use and conversion. And then finally, of course, if we don't use all the CO2, we have to bury it somewhere. And that's another major area of research here in MITI that we cannot cover today. So thank you for your attention. I'll hand over to Mert Yogi.
Well, while this gets set up, I'll share a few anecdotes. When I joined as a faculty member in 2013 to MIT, and it so happened, really to Bob's great credit, and to Mighty's great credit, that my actual first funded grant at MIT was one that was garnered through the auspices of the MIT Energy Initiative. And so in many ways, Mighty helped me launch my independent career. And I think as anyone who remembers the time when they were a junior faculty, that first grant is probably one of the biggest, if you will, hurdles in becoming independent, establishing yourself, developing the sort of confidence to build your own program. And so I can say with a true sort of deep sense of appreciation that Mighty was there with me from the very beginning of my independent career. And I think it speaks to, and I'm not the only one who can speak in a similar capacity on the number of careers that Mighty has helped launch over the years and the technology that it has ultimately spurred. So I'll tell you about that right now, but I want to thank Bob and I want to thank Mighty. Use this. Okay. So the work that Mighty supported actually very early on was our sort of grand vision and dream that it would be useful to be able to use renewable electricity to convert CO2 into value-added chemicals and fuels. And this has been a long journey. It's been something that my group has worked on for the better part of 10 years or so. And I'll give you a little bit of a historical perspective on where we were, where we went, and where we're going. And we do think that many of these technologies could be substantial game changers for utilizing CO2 more efficiently. I should also say one other thing. In addition to Mighty helping me launch my own career, as shown on this slide, the work that was funded by Mighty, that was done as part of this work, has now launched many other careers across the nation. Many of the students who were supported by Mighty in this effort have now launched their own independent research programs in this area of energy, as well as many other areas of energy, and none of that would have been possible without the support of Mighty. So Bob's reach and Mighty's reach extends well beyond MIT's borders through the sort of follow-on effects that we have as academicians and training students. So the vision we sort of had was that you could, in principle, make an electrochemical device that would, on the one hand, oxidize water to oxygen, on the other hand, source CO2 and convert it back into a fuel or chemical that you could use, right? And this sort of grand vision could be appreciated by sort of sourcing renewable electricity to power this conversion, and utilizing CO2, either sourced from a concentrated point source or from one of the many technologies that Alan has developed, to be able to use as the inputs for CO2 to fuel conversion. So again, on one side, you'd be doing oxidation of water. On the other side, CO2 to some desirable product, ideally a chemical or fuel. And it's interesting that the reason you can do this, of course, is that the sites of the oxidation reduction are separated, right? So you don't have a back reaction between the two species. And it's notable that industrially, we're actually witnessing right now a huge growth in water electrolysis technologies, which essentially does the same reaction, but turns water into oxygen and then turns protons into hydrogen on the other side. But the same analogous technology, even though water electrolysis technologies have existed ever since the sort of mid-1800s and have been commercially operated, I mean, alkaline electrolysis ever since the early 1900s, there really had not been any technology development on in the context of a direct CO2 electrolysis system, right? And that, to our mind, was due to many technology challenges as well as many basic science challenges that underpin them. And that's what we started to work on. And again, as a young assistant professor working in an area that was so nascent, I really owe a huge debt of gratitude to Mighty for supporting this effort. So what we appreciated early on was that unlike water electrolysis, where the only reaction you can do to water when you reduce it is to make hydrogen, when you do CO2 reduction, you have the possibility of making a whole host of products. And in fact, you can even do the parasitic reaction you don't want to do, which is the reduction of protons to hydrogen. And so you can make a whole bunch of different things. Now, on the one hand, this is very seductive because it allows you to make a whole host of products that might be industrially valuable, might be precursors to fuels, might be able to be used for a number of different applications. But the problem, of course, is that you can make many, many different things. So you want to make one thing selectively. And it's the science of how to control that selectivity that simply had not existed prior to our work in this area, the work that Mighty supported. And the point I want to stress here is that in principle, the entire carbon biosphere, the entire carbon world we live in, can fit within about negative 0.25 and positive 0.2.5 volts. So a very narrow range of thermodynamic energy accesses the entire diversity of our carbon world. So in the long-term vision where we could make technologies that would be able to select for one product over another, you could imagine remaking the chemical and fuels enterprise through processes like this. We're not there yet, but that's the grand goal that stimulated our interest, right? Okay. So how do we kind of figure, and I'm concatenating many, many PhD theses work into a one slide or a portion of a slide, and also many funded projects from Mighty into one slide, including several Mighty seed projects that got a lot of this work underway. But we learned through a number of studies that my student Anna Wooding, one of my first graduate students, did, was that there was a key kinetic branch point that on metal surfaces that catalyze these reactions, the pathway towards making hydrogen involved critically the combination of an electron and a proton at an interface. This I'm showing just a sort of generic depiction of an electrode surface, but critically that the CO2 activation steps were independent of the protonic environment in the medium. In other words, if you varied the pH or the proton activity or the proton donor, you would dramatically affect this reaction, but not the activation of CO2 and all the downstream reactivity. And this sort of established in our mind, and now actually something that's universally adopted in this community, the basic tenet of how you control selectivity for CO2 conversion, which is to modulate the local environment in terms of the proton activity in donors
in order to be able to select for one reaction over another. So in the very early days, once we understood this, we could actually deploy it very readily to be able to systematically control selectivity. We actually showed that by changing the material structure to be able to change the local pH environment, you could actually systematically toggle the ratio of hydrogen to, for example, CO. We've shown later on you could toggle the ratio between methane and ethylene, between a whole other different products by using this very basic principle of modulating the proton donating environment near your electrode surface. Now, I should say, we did this work, we sort of found this work, and it's incidental that the community, now if you look in this field now, when I started, you would maybe find 1 or 2 papers in this area a year. Now there are about 1,000 papers a year in this area. And if you look at almost all the iterations of people trying to do CO2 conversion, they all apply this basic foundational principle of exploiting the differential and reactivity across different local environments that the catalyst operates in. But as a result of that, the community has appreciated now, just in the last few years, an emerging new problem that this creates, which is that if you have an environment with very low proton activity, so you disfavor hydrogen evolution, unfortunately, since CO2 is an acid gas, the medium you're in actually reacts with the CO2. So while you're able to foster very high Faradaic efficiency, in other words, where the electrons are going, what you suffer is very low carbon utilization efficiency. And that's a big problem because, of course, your carbon isn't free. The CO2 has to be captured, concentrated, cleaned, et cetera. And so to manage the carbon efficiency, you want to inhibit the parasitic reaction of the electrolyte with the CO2. And that's actually what's been sort of appreciated in the last few years. And so very recently, we've sort of addressed this frontier challenge. How can we, at the same time, expose the high selectivity that we understood mechanistically early on, and do it all the while also inhibiting the reaction of CO2 with the actual electrolyte, right? And so recently, in work that was recently published just this year, we found that you could do this if you altered the solvating medium that you were in. The key to why there is that competition is because the parasitic reaction of CO2 and water to make carbonic acids and bicarbonates. So if you went to a medium where you went to a non-aqueous electrolyte, we showed that you could actually preserve very, very high selectivity for CO2 conversion, approaching north of quantitative conversion, in this case for carbon monoxide, but you could do it all the while actually sampling and showing that your medium actually took up very, very small amounts of bicarbonates into the electrolyte. And so it allows us in a sort of rational way to ask, how do you design your environment? How do you design your cell? How do you design your catalyst? All to interplay in the correct way to guide selectivity down the pathway you want and not the pathway that you don't want. And all this might seem very, very fundamental, but I stress that there are now dozens of startup companies and technologies that have been founded on these basic principles, both at MIT and elsewhere. And the work we're doing, I think, can make frontier advances in increasing the efficiency of all of these processes. So I just want to kind of conclude by saying that there is a rich and still largely untapped opportunity to be able to use CO2 as a resource, use renewable electricity as a complementary input to be able to meet diverse needs in chemicals, fuels, and materials. But the key to unlocking all of that revolves around very many basic science questions. And these are basic science questions that encode for how you steer reactivity along one selective pathway over another. And I think MITE's vision, in terms of being able to solve problems, has within it the appetite to be able to support foundational thinking that can lead to game-changing solutions. And I think that's something uniquely a feature that has been supported by MITE and what's made it so impactful on our campus and so impactful outside of MIT. And I can't, once again, thank Bob enough for all the support over the years. I will turn it over.
Right. Thank you for having me. And I have to say, MITE has been a great place for me when I got started. I remember meeting Ernie and Bob coming in as a junior faculty member. And and the kind of support I've gotten has been incredible, both in developing the science, but also translating it. Bob has always been there. I could, call him at any time or send an email. He's always there, and I could go talk to him about things, and has been incredibly supportive. So thank you so much, Bob, and I look forward to you not disappearing away from here, but for your counsel over the coming years. So for me, as I said, I work on interfacial phenomena, and it has been a great journey with MIT because I started with thermal fluid processes, and today I work on electrochemical processes and actually take advantage of both of these to improve efficiency. And today I'm going to, when I started early on, we were looking at flow assurance problems and improving condenser efficiency, boiler efficiency, power plant efficiencies, and to now a lot of work on CO2 and trying to think about scaling that. And these are some of the companies that came out of the lab, and all of these, most of them have a seed fund where a lot of these got started. Liquid Glide to Dropwise, which was a collaboration with Karen Gleason. Infinite Cooling was a Tata-funded project, and so was Axin. So, in my case, all the way from idea to market, MIT has been sort of all along the way. So I'll just highlight few things before I jump into what we are doing on decarbonization. The problem of liquids sticking to solids is kind of a ubiquitous problem. We were looking at actually reducing friction in pipelines and developed a solution. And my wife was asking me to solve a problem on getting honey out of a bottle. And that led to this, where we were looking at how to evacuate containers. And this led off to a company where now this product has been commercialized with Colgate. It's on the shelf. So went literally from idea to the shelf. And this is the world's first fully recyclable toothpaste packaging. And we are doing this across the board. There's a lot of product that's left behind, and believe it or not, we all use consumer packaging. Right? 50 plus billion packaging is made, and the CO2 footprint and the water footprint of the wasted product is kind of enormous. I can go on and on about this. But this is a success story where we started with a seed fund and ended up where this company is going to solve some significant challenges and ensure sustainability of the CPG space, but also others. So I want to talk about what we are doing on the CO2 space. And if I get enough time, I'll walk you all the way through capture to conversion and hopefully to some photobioreactor work. As Alan mentioned, this is a crazy problem, and to be able to get to meet some of our goals, we should be removing 10 gigatons of CO2 must be captured, right? And so the question is, how do you do this? And when you look at this overall value chain, you're going from, let's say, capture to conversion. The capture part is essentially a lot of CapEx. And then as we go to extraction and conversion, significant amount of energy and also CapEx. So the focus of my work has been looking at what are the best ways to do efficient capture. So can there be ways to improve over all the sorbents and things that are out there, and then can we convert them to value products? So if I look at towers like these, which use amines and so on, essentially the capture they're fully soluble, and the capture efficiencies in terms of CO2. Getting CO2 solubility is pretty high, but you still need pretty large towers. And as you go to higher end, as you go to lower and lower concentrations, it becomes even more worse. And so these are some examples. This is a Petronova tower, which is almost the size of the Prudential Tower. Costed like $500 million to build, and then went out of business when they couldn't use the absorbed CO2 for EOR stuff. And so the question was, why do you need such big towers? Because that leads to a lot of CapEx. And so when you look at this, these are all made out of packed beds, and the film thickness is on the order of 600 microns. So if you're able to take that and convert it into finer and finer droplets, your interfacial area increases significantly. And so you can then shrink the size of these towers. And so that led to, okay, if this is such an obvious idea, why didn't anyone do this? The problem is all these fine droplets or mists can also get entrained and become an emission. So thanks to some of the other technology which we had developed to capture plumes from cooling towers, we use some of those ideas to say, hey, we can capture some of these. So this is an example from before, where we were using electric fields to inject charge into these plumes and then are able to capture, and in this case for cooling towers to recycle water from power plant cooling towers. But using similar approaches, we've also taken now CO2-laden mists and are able to do the same thing and capture these. And thereby, what you can do now is go to much smaller drop sizes and have a capture unit like this that can lead to large capture efficiencies, but also larger CO2 capture efficiencies. So here is a conventional bed, and we can significantly shrink the size of these and so reduce the capex. And we can also open the door to other solvents in this process. If you take DAC, also 60% of the cost is capex. So a significant cost of the levelized cost of CO2 goes into capex. So some of this work is we are trying to essentially improve the capture efficiency. And we know we have done this already with cooling towers, where we can scale this technology. This is an implementation at the MIT cooling tower or at the Nuclear Research Lab. And this is at a gigawatt scale power plant where we are capturing water in this case, but then in the future could be used to do capture of CO2 from these. The other project, where capture has been capture is already done for us, and this is in collaboration with Alan, who's a dear friend and who has come up with a lot of the interesting ideas around this, where we are making some interesting advancements. Is CO2 capture from oceans. The concentration of CO2 is 100 times more in oceans. So looking at the carbonate bicarb equilibrium that Alan had talked about before, by adding protons here, we can actually release the CO2. And so what has been done in this project, and we continue to work on, is have two cells where we acidify the ocean water, release CO2, and then we can realkalize. And this system, as we have it, has one of the best energetics in the world today. And we have no waste products, all done with electroactive materials. Now, if I have, Alan, do you have 2 minutes? Or yeah, I have 2 minutes. So I'll jump on the conversion part, which is, I think, a important thing, as Yogi stressed. And one of the aspects that I don't have to do the introduction because Yogi has done it so nicely. One of the key challenges here has been hydrogen coevolution. And I believe aqueous solutions are a way to scale. But then we had to look at how to resolve this co-evolution of hydrogen. Because as we go to looking at products, if I go to ethylene, we want to be at very, very high current densities on the order of amps per centimeter squared to make it economically possible. And if we want to achieve that, a key problem is hydrogen coevolution. So GDE architectures have evolved over the years. And a key challenge has been a trade-off between sort of conductivity and wettability. If you are a carbon electrode, you can get really good conductivity through the thickness. But then what happens is it can flood and you lose and you introduce sort of a lot of mass transport limits. If you are a PTFE electrode, you have good hydrophobic properties. You don't flood, but then your conductivity is limited to only a plane. And so these are some things. What happens is, as you scale such electrodes, you are significantly limited both on the amount of overpotentials as well as the loss in the product. So we sort of were able to combine some of these ideas at a system level and come up with sort of hierarchical conductors that goes into this space where we can sort of meet the trade-off between both high conductivity and high hydrophobicity. And so this is also showing how we can manufacture these by just weaving techniques and so on. And we have been able to go actually scale up from these sort of lab scale 5 centimeters square to almost an order of magnitude with high Faradaic efficiency as well as selectivity. And for these ePTFE electrodes, we've been able to show that we can really push this and move towards scale. With that, I'm going to stop. And one last thing I wanted to point out is if you're able to integrate, as a lot of people are trying to do, you can, by reducing CapEx and the energy cost, we can potentially come up with an architecture like this where we could, capture these mists, do the conversion, and regenerate our electrolyte. So with that, I'm going to stop. And I know Chris and Angie will talk about some of the bio conversion approaches.
Thank you. I'm very excited and honored and a little bit sad to be here today, but definitely excited to be involved in this celebration. I've been thinking about my interactions with Bob since before I was a professor here, actually, when I was still visited with him when I was still a postdoc, and thinking about how important that he's been to my career. And when I was putting slides together, I was really realizing that almost everything I've done has been touched by Mighty, and I'll show that in a little bit. But I was thinking about just what a huge support Bob has been for my entire career. And it's not just the science. And so not only are you an amazing engineer, but your ability to understand how to interact in so many situations. I'd come in here and I would be like, okay, I have this situation. And Bob's like, okay, let's just think about this and work through it. And by the time I left, it was like I knew how to solve the problem. And yeah, I think you taught me a lot about just, being a professor and being an engineer. So with that, I'm very honored to be at this celebration. So it's been my whole career, pretty much, I've been fascinated with how to give organisms the ability to work with non-living structures, how to genetically encode non-living structures, and that has gone through my work, my Mighty work as well. Anything from building virus-based batteries to genetically engineering organisms to hold on to carbon nanotubes better and give them a second gene to be able to build titania to increase solar cell efficiency. And so I've always been interested in, how do organisms in the ocean make materials? What can you learn from them? How can you take the genes that they already have that code for the ability to do things like make silica-based materials or calcium carbonate-based materials, and how can you give them the opportunity to work with the rest of the periodic table? And that in my lab, we work on simple organisms. We work in bacteriophages, which are viruses that infect bacteria. We work with bacteria. We work with yeast, and now with some other kind of fungus as well. And the funding I had from Mighty has been is how do you coax biology to work with electronic materials and incorporate multiple electronic materials together to give new kinds of functions? And if this works here, yeah, the idea here is we all know viruses. Bacteriophages are a kind of virus that infects bacteria. How can you take an organism that evolves very quickly, a simple organism that only has a couple genes that code for a couple of proteins, and through a combinatorial library approach, find the right genes that code for the right proteins to make new kinds of materials? And that's something that Mighty has supported me on the whole time. This is the periodic table. Only a couple of elements in the ocean where organisms came from. Organisms use. They use calcium carbonate, they use calcium phosphate, they use silica, they use iron, but most of the elements are not used by biology. And what my lab has done is convince organisms to work with the rest of the periodic table so that you can have a gene for the ability to make a material for a battery or a solar cell. So when I was putting, so this is a picture of one of my best collaborators, Professor Paula Hammond, who we all know. We've had collaborations with MIGHTY for giving DNA sequences to the ability to grow better solar cells, to grow better batteries, better catalysts. I've spun out 5 companies based on this technology over the years. One in catalyst, one in materials for fuels, one in environmental remediation, and 2 in cancer. And all of it has come from this basic idea of developing biology to grow new kinds of building blocks. And we've also worked in artificial photosynthesis as well. So this is actually a list of, we've made up 110 materials that are biotemplated. That means they have a DNA sequence that codes for a protein that can grow these materials. And if you look at these materials, what you'll see most of these are not things that would normally be grown by biology, but you can actually find the right protein sequence to be able to control them at the nanoscale. So to be able to nucleate the material, control its architecture, control its nanoscale geometry to try to get increased function. And so one of the first topics that we worked on through MIGHTY was in batteries and energy storage. And at first we started working on materials that were, I'll show a little bit about them, but we've gone through multiple scales now, anywhere from small batteries to batteries that are structural batteries made for UAVs, to batteries that are integrated into textiles, to lithium oxygen batteries for vehicle applications, and are now working on grid and microgrid applications. And the idea was to be able to find a virus that could do multiple things: pick up a carbon nanotube, which is a good electrical conductor, grow a good material that's a good ionic conductor, wire itself to the current collector, and then self-assemble into a battery. And that worked with many different kinds of materials for regular lithium-ion batteries. It worked with lithium oxygen battery electrode materials. And we are using the power of genetics. We're using the power of evolution. So you always have the ability to evolve it to become better. And then we've also used it recently in making structural batteries, which are integrated into UAV applications. And this is a company I founded, Siluria Technologies, that was using biology to make new kinds of catalysts and catalyst combinations for natural gas to ethylene, ethylene to drop in replacement fuels. Through our work with MIGHTY, we've also worked quite a bit in environmental remediation, clean mining. We had a failed project in enhanced oil recovery, where we're using biology to make really high surface area carbons on the order of 2,000 meters squared per gram. That work failed. We tried to use it to make lithium sulfur batteries. That failed. But what we actually found was that we could make this really high surface area materials that was really good at neutralizing chemical warfare agents, soman and sarin. We went on and scaled that with the Army in the multi-kilogram scale to where they were making 2,000 liter reactors that they now have that you can use shampoo for neutralizing chemical warfare agents or bad industrial chemicals. That's not something we have to worry about in our lab. So we then repurpose that material to clean up PFOS, forever chemicals, to make a super high material that was very absorbent of PFOS and different combinations of those, but then could also have catalysts in it for breaking it down to non-toxic materials. And that's a spin out of my lab called 6x6 Biotechnologies. Another project that we worked on was, can you genetically engineer yeast to clean up very toxic waste? Sounds like a kind of crazy idea, but people have been engineering yeast for 5,000 years. And so can you use them to deploy at scale for cleanup? This is a work that we did from the oil sands in Canada, where we genetically engineered yeast to be able to clean up copper, lead, mercury, zinc, and cadmium, go from water like that, or oil sands like that, to drinking water after 3 or 4 rounds of selection and evolution. and it's an active area of our group right now. We're moving that to mining, clean mining, mining runoff. The last thing I want to say was that if I hadn't done any of this, started making these materials for catalysts, solar cells, and batteries, I wouldn't be working in ovarian cancer now. And I have a 10-year plan to solve ovarian cancer, along with Paula Hammond and Sangeeta Bhatia. And what we did was use some of the same materials we developed for batteries and solar cells and started repurposing them to find tiny tumors for ovarian cancer during surgery. And from there, we're moving on to the idea of diagnosing ovarian cancer at least 10 years earlier, with the idea of having ovarian cancer a disease of the past in the next 10 to 15 years. So, Bob, you're amazing. You've been a great mentor. Mighty is amazing. I feel so lucky to be have been in this group of amazing scientists and engineers and, well, everyone since I pretty much first arrived. And I know I wouldn't have done anything that I've done without the support of Mighty. And I definitely wouldn't, have found these companies and be on a mission to cure ovarian cancer. So thank you.
I have to be careful. I notice if I stand too far that way, I blind myself. So I don't think I've known Bob the longest of the people on this panel. I do think I was the youngest when I first met him of the people in this panel. I believe I gave my first presentation in front of Bob 30 and a half years ago as an undergraduate student. And in the old 1027 now is a course number we use for our energy projects lab, but it used to be our unit ops lab. And one of my 3 unit ops projects was one that was run by Professor Armstrong. We didn't call him Bob then. He was the esteemed Professor Armstrong. I remember giving a very important and nerve-wracking talk 10 years later, so 20 and a half years ago, rather than 30 and a half years ago, when I was a faculty candidate, and Bob Armstrong was the department head. And I'm sure I gave a talk 10 and a half years ago to Mighty and something. But there are lucky threes here in 93 and 2003 and in 2023. And so I have to echo the comments of everyone in talking not just about the science, but in the influence Bob's had as an individual. I'll tell one story that I don't have a slide for. When I interviewed with MIT, I remember Bob asking me, when we talked about an offer, what are you afraid of? What's the thing that is most concerning to you about being a professor at MIT? And I didn't have children at the time, and I said, I really want to have a family, and I just don't know if that's possible to do this job. And so he told me a story, I don't know if you remember telling me this story, about how when he was a young faculty member, he was a little league coach for his son's team. And when he came into the office on game days, he was wearing a baseball hat. And the baseball hat was a signal to his admin that come hell or high water, he had to be out of the office. Was it 3:30? He had to be out of the office at 3:30 because he had to be there to coach his kids' Little League games. And I remember going home and telling my husband, I think I can do this because I think there's a department head who will support my desire to do this. And the transformational impact that had on me as an individual is very hard to overstate, but it really has had a transformational impact on our department. So I stand here as a member of the Department of Chemical Engineering, which is filled now with women faculty member. The vast majority of us have children, who had children prior to tenure, and who knew that we could do it because of the standards and the examples that were set by a transformative department head. So thank you for that. Now I'm going to talk about science. But I would be remiss if I didn't start in talking in that way. The work that I do, and I've sort of made this joke, I told someone, I keep getting asked to talk about energy, and I don't do a lot in energy, but I do a lot in energy. It's just not, I would say, the traditional energy. So what I want to talk about is the work that I'm doing, but also link that to the foundations in Mighty, but also to this grander problem that we have to solve in climate and sustainability. And that's come across many times, including from Ernie's remarks and from others on this panel as well. I want to start by looking at a barrel of crude oil. And you can read the numbers. I'm not going to read them for you, but I'll say that the bulk of this is an area that we're accustomed to thinking about and accustomed to talking about, which is if we're using that material for energy, where are we going to get alternative energy from? And I used to have an old picture that would go there that showed an ear of corn going into the into the tank. And so the early work that I did with Mighty was very much focused on how to actually bring biofuels to market and really think about designer molecules. I'll talk about this a little bit more, a little bit later. But how do we bring molecules that are purpose-built for the purpose of being used in internal combustion engines, but to be produced from biofuels? I think since that time, the consensus, which I do support, is that we can actually rely upon electrification for light-duty vehicles, for things that are on the road. I have gone on record as saying I am not flying a solar-powered airplane across any ocean. Right, so I still think when we talk about aviation fuels, we're going to need that. But by and large, the movement towards electrification has taken a lot of the focus around the production of biofuels for vehicles, for transportation in terms of on the road. But there's another segment which is very interesting from my perspective as a chemical engineer, and this is where the material in that barrel of crude oil is not being used for energy, not for the energy in those chemical bonds, but being used for their material properties, for the mass. And when we look at that, that's not something that you're going to replace with nuclear power or with wind power or solar power, but you actually have to figure out where you're going to get access to the physical materials that you need. And that's been talked about already in this particular panel. The focus that we have is actually connected to what Angie just talked about, which is the power of being able to utilize biology for this. And we're motivated by what a colleague of mine at MIT, Kimmy Brad-Olsen, described as a paradigm shift, meaning that we need to go from a reliance upon hydrocarbons to one that relies upon carbohydrates. And those carbohydrates can come from sustainable, renewable biomass and provide us with sugars and other simple feedstocks that we can use, yes, to make biofuels for very specific purposes. I've already mentioned aviation as one place where this still makes a lot of sense, but also to start thinking about what are the chemicals and materials and what are the industrial processes that go into the production of those chemicals and materials that might be altered or modified and substantially improved in terms of their energy efficiency by looking at alternative sources. So I mentioned we do this using biology. I give a talk and I'll say when I was a chemical engineering undergraduate, this was the view of the cell we were given, which was don't worry about what happens inside of it. It's just a thing and you're going to work outside of it. But the cell is really just a collection of lots and lots of chemical reactions. So our ability to be able to take a biological system and use it to make new molecules comes from the innate ability of biology to do this anyway. We have proof that you can take simple sugars, you can take in many cases oxygen and nutrients, and make a wide range of materials, including drugs. These are all natural products that have been produced, nutraceuticals, but also industrial chemicals. Okay? And so what we've been able to do then is to take this philosophy, if you will, and use it to be able to tailor make specific molecules for particular purposes. So I have three quick examples. One, that's a compound glucaric acid I'll come back to, which require 3 genes from 3 different organisms, a yeast, a mouse, a bacterium. I used to give the joke, it sounds like the beginning of a joke, a yeast, a mouse, and a bacterium walk into a bar. But I could never figure out the punchline, so I stopped telling the joke. We use it to make a compound that's actually used as a pharmaceutical precursor. And in a Mighty funded project, we actually used it to make a bespoke compound that was requested. Can you make something that has 5 to 8 carbons, that has 1 oxygen, that has 1 branch point, because of research that had been done to determine that it would be an ideal kind of biofuel. All along the way, as we leverage the power of biology to do this, our goal is to be able to make things that you can't make in other ways, but also to make sure that we're focused on accessing molecules that we can produce economically. We know that there is an imperative to be able to displace the use of fossil feedstocks, but we also have to be mindful of the realities of economics. Our overwhelming goal or overarching driver for this, though, is to reduce the imprint on the footprint on the environment and to have increased sustainability. So I do want to walk through 3 very quick examples. Others have talked about innovation and entrepreneurship that has come out of this. I'm a co-founder of a company called Calion that has been commercializing glucaric acid production. And I'll say, Angie ended her talk with this vignette of all these things that didn't work, but then ultimately work. And this is an example of a compound that we had some ideas about how it might be used, but it's turned out it's really very attractive in the context of water treatment. And one particular example, here's a picture of a cooling tower. All the engineers in the room, chemical engineers in the room, are very familiar with cooling towers as essential pieces of unit ops that are there to allow you to regulate the temperature of water. The water that you use, or temperature of processed fluids, the water that you use in cooling towers, though, has to be treated before it's released into the environment. And the traditional way to do this is with phosphates. The problem with that is if those phosphates end up in the environment, they actually can cause algal blooms because you're now providing a nutrient that's naturally present in very low concentrations. Once those algae, which are starved, begin to consume it, they actually grow to the point where they block out the sunlight and they kill the aquatic life underneath it. So Europe has actually been looking to remove phosphates from wastewater treatment. Turns out glucaric acid can be used for this very well, have none of those impacts on the environment. And some back of the envelope calculations from the company have actually shown that you get improvements in efficiencies of the operation of those cooling towers, reducing fouling. And in doing so, the estimates are that you can reduce about 230 million metric ton equivalents of CO2 per year by switching over from phosphate to this. And that could result in global savings about $60 billion in terms of efficiencies in the system. So again, thinking about a very simple compound, but the impact it has on industrialization. The second example I want to give is about agriculture. And this is the subject of a collaboration. Ernie talked about the significance of collaboration within Mighty, and that's been shown in a lot of our work as well. I want to draw your attention here to the fact that about a quarter of global greenhouse gas emissions is actually related to agriculture. A lot of that is about the production and dissemination of fertilizer, in particular nitrogen-based fertilizers. And about 30% of energy use in the U.S., on farms in the U.S., is related to fertilizer. This is actually a collaboration, as I said, with Chris Voigt, where the idea is to be able to take advantage of bacteria that will normally fix nitrogen from the air, provide that into crops, and the crops will make then the sugars that feed the bacteria. This is true for legumes. It's not true for cereal crops. And it turns out we actually need more cereal crops than anything else to be able to feed the planet. So the idea here is to be able to actually engineer microbes that can affix to the cereal crops with the same ability to fix nitrogen and allow you now to dramatically reduce your dependence on externally applied nitrogen. This is actually the technology that has been commercialized by a company called Pivot, which was co-founded by Chris Voigt, that leverages technology from MIT. The Climate Grand Challenges project actually says, can we take that further and just engineer those plant crops directly to be able to fix CO2? So again, a lot of these principles that came out of can you engineer microbes in order to make biofuels are now being used to translate into can you use them to solve bigger problems. Very quickly, the last example I want to give is around plastics. You can see the numbers that are here. The main point to make here is about 80% of the plastics that are produced actually end up accumulating in landfills or in the environment. So this is a collaboration with Brad Olson, with Desiree Plata, where the idea is to be able to design plastics from the beginning for degradation, but also to be able to exploit monomers that can produce to make those plastics from biological resources. The key thing that we're working on now is to be able to prove that it actually is biodegradable and to be able to develop the assay and analytical methods to be able to reduce those screening times. The second aspect of this is to be able to really use complex microbial communities to more effectively mimic what will be seen out in the environment. So I will just end by thanking Bob for the person that he is, as I started with, but also really the transformative impact that he's had. And I hope that in this talk, what you could see is how the seeds of Mighty have actually started to have a much broader impact across the campus in a lot of our climate and sustainability efforts. Rob is standing, so I will stop talking. Thank you very much.